In the last classes, we discussed about plant relations, water plant relations. In the second class, we discussed about mineral nutrition, wherein we discussed about the importance of nutrients for the healthy growth of a crop or a plant, and the classification of various nutrients as macronutrients and micronutrients and their deficiency symptoms, etc. And in the last class, we discussed about enzymes. What are enzymes and how the enzymes are named, or the nomenclature of enzymes, and the latest mode of classification of enzymes, and how do enzymes work, and what is enzyme inhibition, etc. we discussed. And in today's class, we discuss about the photosynthesis. You know, photosynthesis is the most important biochemical reaction that is occurring on this earth. It is the most important. Why I use the most important? Because it is because of photosynthesis, the food is manufactured or prepared, and this manufactured or the plant manufactured food on which all plants, all animals, all organisms as such depend directly or indirectly on the photosynthate, what we call. The end product of photosynthesis is called photosynthate, that is nothing but your glucose or starch. Now, the meaning of photosynthesis, photo means light. Synthesis means it is an anabolic process wherein, wherein small energetic molecules are added, simpler molecules are added into a complex energy-rich molecule, wherein here the simplest molecules are carbon dioxide, water, energy, that is light. So carbon dioxide, water, in the presence of chlorophyll, by using the light energy, these simpler molecules are converted into complex energy-rich molecule, that is nothing but your glucose or the starch. So this is what we call photosynthesis. So this photosynthesis otherwise can be defined as conversion of light energy into chemical energy. So the source for the entire globe is sun. The entire source of energy for this entire globe is sun, and whatever the energy that we get from sun is converted into different forms. So the conversion of light energy into chemical energy is called photosynthesis. So photo means light, synthesis is energy, formation of energy-rich complex molecules or compounds. Now this photosynthesis is done by plants only, because all green plants which have chlorophyll in them are capable of converting this light energy into chemical energy. So these green plants are only capable of converting this light energy into chemical energy. So the exception, all plants, exceptions, the fungi and the parasitic plants. These two groups of plants are exceptional in their heterotrophic behavior. Except that these two groups, all plants are autotrophic in nature. That means they prepare their own food material. So this process of conversion of light energy into chemical energy by green plants or chlorophyll containing plants is called photosynthesis. Now this photosynthesis, for our convenience, we divide this photosynthesis into two phases or two parts. One, what we call light dependent part. The other is light independent. So first phase is called light dependent. This is otherwise called light reaction. And the second one is called light independent, or otherwise called dark. See, don't get confused. Dark reaction means it doesn't anything related to darkness. So it means it doesn't need any light. It utilizes the products of the light energy or the products of light phase. So the end products of light phase, that is ATP and NADPH2, are utilized during light independent phase or dark phase to convert this NADPH2 and ATP into carbon or carbon rich, energy rich carbohydrate. So again, the photosynthesis for our convenience is divided into light phase and dark phase. Now we confine ourselves in today's class to light phase only. So this light phase is light dependent. So this light phase occurs in chloroplast. Of course, both the phases, light phase and dark phase, occur in the chloroplast only. So the light phase occurs in chloroplast. I think the structure of a chloroplast. Plants differ from animals in having chloroplast. Chloroplasts are absent in animals, hence they are heterotrophic. They depend on plants directly or indirectly for their nutrition. So, the photosynthesis equation, that is 6CO2 plus 12H2O in the presence of light, the chlorophyll molecule gives rise to what we call C6H12O6 plus 6H2O plus 6O2. This is the basic equation or the fundamental equation of photosynthesis. Though it looks so simple, but it is not so simple. It requires hundreds of enzymes for the ultimate production of end product. So generally, the carbon dioxide and the water, they are called reactants. And the end product is glucose. And of course, byproducts are water and oxygen. So now, this glucose is the primary product or the fundamental end product of the photosynthesis, on which, or it is the primary product which gets converted into other forms. See, first energy-rich compound that formed on this earth is only glucose. And this glucose gets converted into different forms in the presence of enzymes through various biochemical processes. So from this glucose only, our lipids, our proteins, our enzymes, our hormones, so what not? Your nucleic acids, everything are formed from glucose only. So hence glucose is the primary or the fundamental metabolic compound. So now, of course, first of all, begin with the structure of chloroplast. Chloroplasts are of different shapes and sizes. Generally, in algae you studied, there are various shapes and sizes. But of course, if you take a cell, for example, a plant cell, now this chloroplast is very big organelle, cell organelle. This is the biggest cell organelle of the cell. Now, these chloroplasts are of various shapes. Mostly they are ovoidal, egg-shaped. And you find four important components or important parts in a chloroplast. One is the membrane, a double membrane envelope. And inside this double membrane envelope, there is a gelatinous substance which is rich in various enzymes, DNA, RNAs, many things are present inside this viscous amorphous substance called stroma. This is the second part. Embedded in the stroma, you will see many thread-like structures called lamellae. These lamellae are otherwise called thylakoids. And these thylakoids, they are seen traversed throughout stroma. And here and there, the thylakoids, they form certain disc-like structures. They form piles of a coin-like structures called grana. So in the stroma, you will find many coin-like structures called granas, and such granas are connected by thylakoid membrane. The thylakoid membrane that connects two grana is called a stroma thylakoid. And the thylakoid that is present inside the granum is called a granum thylakoid. So thylakoids have two types. One is granum thylakoid, the other is stroma thylakoid. Now if you observe the complex substances that are embedded in a thylakoid membrane, you will see. So by subjecting this, thylakoid to mild detergents and doing ultracentrifugation, scientists were able to isolate 4 important complexes from thylakoid membrane. Those 4 important complexes are photosystem 2, cytochrome BF complex, photosystem 1, and the other is ATP synthase, or otherwise called coupling factor. So these are the 4 important multi-protein complexes embedded within the thylakoid membrane. Now we'll study in detail one by one. So now these 4 complexes are present inside what we call thylakoid membrane. So now if you want to know more about these complexes, we should know about the pigments, the important pigments that are present inside the granum. So inside the granum, you have pigments. The pigments are responsible for absorbing the radiant energy from the sun. Now these pigments are called, there are 3 major types of pigments. They are what we call chlorophylls, carotenoids, and the other is phycobilins. Chlorophylls, of course, chlorophylls are many a times. So now you studied in your algae, based on the nature of the, coloration, algae are classified into, 8 to 9 classes: chlorophysic, xanthophysic, rhizophysic, bacilharophysic, cryptophysic, dinophysic, etc. So in that classification, the pigmentation is one of the criterions. Most important criterion was type of pigment that is present in that particular algal group. Now these chlorophylls, chlorophylls are the primary pigments that are responsible for absorbing light. The light, it is absorbed in the form of what we call photons. Now this chlorophyll, chlorophylls are again classified into 6 to 7 important types. Of course, some people say 9 types of chlorophylls: chlorophyll A, chlorophyll B, C, D, E, bacterial chlorophyll, and chlorobium chlorophyll. These are the 9 different types of chlorophylls. Now chlorophyll A, this is what we call universal pigment, present in almost all plant groups. Chlorophyll A. Now chlorophyll B is the second type. Now this chlorophyll B is present in all higher plants and also present in chlorophyceae. Chlorophyll C is present in phaeophyceae and bacilharophyceae. Chlorophyll D is present in rhodophyceae, and chlorophyll E is present in xanthophyceae. And bacterial chlorophyll the chlorophyll that is present in bacteria is quite different from the chlorophyll that occurs in higher plants or algae. The chlorophyll that is present in bacteria is called bacterial chlorophyll. This is again chlorophyll A and B. So this chlorophyll that is present in bacteria is called chlorophylls of photosynthetic bacteria. Bacteria are of two types, photosynthetic bacteria and of course chemosynthetic bacteria, and green sulfur bacteria, purple sulfur bacteria, etc. based on the type of chlorophyll that is present in them. So like this, chlorophyll pigments are in all classified into 9 groups from chlorophyll A to chlorobium chlorophyll. Now chlorophyll A is the most important and the primary chlorophyll pigment present in almost all the autotrophic plants. Now its a structure, it has what we call, it's of course, its formula is C55H72O5 N4Mg. If you observe, of course, in your textbook, you can see the chlorophyll structure. It has a head and a tail. The head has what we call 4 tetrapyrrole rings, and for the 4th pyrrole ring, you have a long tail, a phytol tail. Now, chlorophyll A and B, these two are primary pigments that are responsible for absorbing radiant energy. Now, this chlorophyll A and B differ in only in the 2nd pyrrole ring. Of course, if methyl group is present, that denotes it is chlorophyll A. If the methyl group is replaced by an aldehyde CHO group, that becomes your chlorophyll B. Now, these 4 pyrrole rings, tetrapyrrole rings, are joined by a magnesium atom in the center. So this is the structure of a chlorophyll molecule. Of course, then we move to 2nd group, that is carotenoids. The carotenoids are again divided into carotenes and xanthophylls. Carotenes and xanthophylls. Now, the carotenoids, the carotenes, they are orange, red in color, and they have what we call only carbon and hydrogen, no oxygen is present in, carotenes. They are the pigments that contain only carbon and hydrogen. And these carotenes are again divided into 3 types: alpha carotene, beta carotene, and gamma carotene. Of course, xanthophylls, they contain carbon, hydrogen, and oxygen, of course, and they are yellowish in color. And generally, the important xanthophylls that are found in plants are, they are lutein, zeaxanthin, and viloxanthin, etc. So this is about the chlorophylls and carotenoids, carotenes and xanthophylls. Then coming to the 3rd group, that is phycobilins. So phycobilins differ from chlorophylls and carotenoids. They are water soluble. They are the water soluble pigments, and they are present only in 2 groups. One is cyanophyceae and also in rhodophyceae. Hence, they are called cyanophycean phycobilins, rhodophycean C phycobilins and R phycobilins, what we call. C phycobilins, R phycobilins. C denotes the class where they occur. C is cyanophyceae, or is rhodophyceae. Now, these phycobilins are of 4 types. One is phycocyanin, which is blue-green rather, and phycoerythrin pigment, and the 3rd one is allophycocyanin, and the 4th one is what we call phytochrome. While 3, that is phycocyanin, phycoerythrin, allophycocyanin, they participate in or they are involved in absorption of radiant energy. And of course, phytochrome is also active, but its primary function is metamorphosis, or what we call, it is what is a photomorphogenesis. Photomorphogenesis, what we call flowering hormone. Then we study about nature of light. A light, for our convenience. So light ray, if you a light ray is passed through a prism, 7 colors. That is otherwise called VIBGR: violet, indigo, blue, green, yellow, orange, and red. Now this VIBGR, its wavelength ranges from 400 to 700 nanometers. Now the wavelength below violet is called ultraviolet, and above red is called infrared. Now you have to remember one more thing. Now this energy and the wavelength are inversely proportional. Lower the wavelength, higher the energy. Higher the wavelength, lower the energy. Thus, violet has lower wavelengths or less wavelength, and its piercing ability, they are highly energetic. Whereas red, the wavelength is more, and its piercing ability or energy content is less. Okay, so now you may ask me why we should know about different these colors. All the colors VIBGR, we divide them based on their wavelength. Now you will appreciate that all colors are not active in different wavelengths. So violet absorbs only light of 380 and 450. Blue, of course, 450 and 495. Of course, green 495 and 570 nanometers. And red, of course, 620 and 750 nanometers. That is, in between two extremes, violet and red, we have different wavelengths. And each pigment or these different chlorophylls have what we call absorption spectrums higher at different wavelengths. Say, for example, your chlorophyll A. Chlorophyll A, its absorption peaks are, one peak is at 435, the other peak is at 673. And chlorophyll B, its absorption spectrum is 453 and 642. That means at 453 nanometers, its absorption is more, and so also at 642, its absorption is more. And chlorophyll A, its absorption peak is at 435, and another peak is at 673. So that means different pigments absorption spectra are different. That means all the pigments, they are active at different wavelengths, and all this energy that is trapped, they are called antenna pigments, and whatever the energy that is trapped will be in turn through resonance will be pumped to active center or reaction center of photosystem 2 and photosystem 1. Now the mechanism of photosynthesis, how this actually the photosynthesis takes place or occurs. Now the earlier researches were done by Robert Hill, Reuben Common, and others worked on this, Arnon. And Robert, especially, we should remember about Robert Hill. Robert Hill, during 1939, he isolated these pigments. He isolated various pigments, that is, various chlorophylls were isolated. Chlorophylls, carotenoids, xanthophylls were isolated. And when monochromatic light was given to that isolated pigments, in the presence of certain what we call electron acceptors, they were successful in releasing oxygen. So this reaction wherein the pigment's ability to oxidize water and giving rise to or reducing certain compounds and ultimately liberating oxygen is what we call Hill reaction. See, for our convenience, Hill reaction, for example, 2H2O plus 2A in the presence of light and chlorophyll, you get 2AH2 plus O2. A means here a reagent or Hill oxidant. That is, this oxidant, of course, it oxidizes water. By how? By taking hydrogen from water through what we call, so in the presence of light, water breaks. Breaking of water in the presence of light is called photolysis. As a result of photolysis, you get electrons and protons. Now these protons and electrons combine with A and ultimately reduce that into AH2. So they have used what we call ferrocyanide. Ferrocyanide. So the ferrocyanide got ultimately into reduced form. And some other people working with, a bacterium, sulfur bacterium. This sulfur bacterium, they don't use water. So instead of water, they use H2S. So when H2S was given, sulfur was given out as a end product, byproduct. So here also H2S plus 2A, you get what we call 2AH2 plus 2S. So if water is the compound that is subjected for photolysis, you get oxygen as an end product or byproduct. If it is H2S, you get sulfur. So because sulfur bacteria, they oxidize H2S into sulfur. This is what we call Hill reaction. So Hill reaction is nothing but Robert Hill said that whatever the oxygen that is liberated comes out from water. It doesn't come from carbon dioxide. As a result of photolysis of water only, you get oxygen. Okay. So now, after that, Emerson enhancement effect. So Emerson working on chlorella, and this chlorella was given a monochromatic light of red wavelength, and he measured photosynthesis rate. When red light was given, there was a drastic reduction in photosynthate. So when smaller wavelength light was given, some amount of photosynthate was produced. When red light was given, there was a reduction in photosynthate production. And Emerson, to his astonishment, when both the wavelengths, when both higher wavelength and lower wavelength were given simultaneously, there was an enormous increase in the photosynthetic yield. This yield with the simultaneous giving of higher and lower wavelengths simultaneously is called Emerson enhancement effect. Through Emerson enhancement effect research, it became very clear that the pigments, some pigments are active, absorbing lower wavelength light. Some pigments are active in absorbing higher wavelengths. That means when you are giving only shorter wavelength, only one type of pigments are active. When you are giving higher wavelength, the other type of pigments are active. When you are giving both simultaneously, means both two types of pigments or two types of what we call photosystems work simultaneously, and ultimately whatever the photosynthate is more than the sum total of lower and higher. Hence, we call this phenomenon as Emerson enhancement effect. Emerson enhancement effect. So through this Emerson enhancement effect only, we came to know that there occurs two photosystems, one being operated by shorter wavelength, the other is operated by longer wavelengths. When you are giving both shorter and higher wavelengths at a time, both the photosystems are operating simultaneously. Hence, the photosynthetic yield is more. Hence, the term Emerson enhancement effect. I think you got this. This is very important in your examination. They may ask you under short notes, Hill reaction or Emerson enhancement effect. Now, we'll study about what we call Z scheme, or what we call cyclic photophosphorylation or acyclic photophosphorylation or proton translocation, etc., etc. Initially, people used to explain these light reactions in terms of electron movement. But recently, of course, not recently, from 1960 onwards, is because of Peter Mitchell and others' thorough research, we came to conclusion that it is not actually electrons, it is actually the proton movement that is responsible for production of ATPs. That ATP production is what we call photophosphorylation. Photo means light. Phosphorylation means production of phosphates. That is, in the presence of light, when ADP plus inorganic phosphate couple with each other and ultimately resulting in ATP, an energy-rich phosphate. Photophosphorylation. So this mechanism of how photophosphorylation occurs and how this reduction of coenzymes takes place was worked out by so many people across the world, and ultimately it is the Peter Mitchell's chemiosmotic concept is currently accepted across the world. Before that, whatever the jet scheme that was given by Hill and Bendall, but of course, still it holds good, but so many people added to that, and ultimately, of course, this Z scheme is modified to suit in the light of present investigations. So what you have to, this is very, very important as far as examination is concerned. They may ask you the electron transport mechanism or proton translocation or Z scheme, etc. Or cyclic photophosphorylation or non-cyclic photophosphorylation. Cyclic photophosphorylation means the electron gets ejected and passes through various electron carriers and ultimately reaches back. This is called, as it is making a cyclic movement, it is called cyclic photophosphorylation. Whereas non-cyclic photophosphorylation, wherein the electron that gets ejected from photosystem to never comes back. It ultimately moves through various electron carriers from quinones to cytochrome B, cytochrome F, plastocyanin, and photosystem 1, then ferredoxin, ultimately. So, by passing through various electron carriers, it ultimately reaches to ferredoxin, and ultimately ferredoxin gets reduced to reduced form of ferredoxin. That is, reduced form is nothing but NADP gets converted into NADPH. And whatever the energy that is present in the protons gets transformed or is used in the coupling of ATP and inorganic phosphate and ultimately giving rise to ATP. This is called coupling factor or ATP synthase. So the energy for the ATP synthase comes with the movement of electrons and protons. Of course, more specifically, protons. The energy-rich protons try to escape through membranes across gradient. So generally, diffusion always moves from higher to lower. But here, the membrane, the membrane does not allow for the movement of, protons. It is impervious. It does not allow. So then there is a mechanism, there is a channel for the movement of these energy-rich protons from the lumen of the thylakoid into the stroma. When the protons move from, lumen of the thylakoid into the stroma, and whatever the energy that is present will be sufficient to couple ADP plus inorganic phosphate, giving rise to what we call ATP. This is what we call ATP synthase or coupling factor. Now we will study this. Now, there are four, in this Z scheme, or what we call acyclic photophosphorylation or non-cyclic photophosphorylation or proton translocation. There are four important, multi-protein complexes. Multi-protein complexes are present. One is photosystem 2. Second one is the cytochrome BF complex. And third one is photosystem 1. And the fourth one is coupling factor, or otherwise called ATP synthase. We'll study one by one. So now, first of all, we start with photosystem 2. Now this photosystem 2, it is a multi-protein complex. Of course, there are 6 integral polypeptides are there. One is an integral polypeptides. Second is reaction center. That is, reaction center is 680, P680 otherwise called. And of course, there are 2 important polypeptides, that is D1 and D2. And there are other polypeptides which are involved in, calcium and chloride attachment, and which help in photolysis of water. So now, first of all, and this reaction center, what we call photosystem 1's, photosystem 2's reaction center, it has what we call, it has some 40 chlorophyll A molecules and 25 chlorophyll B molecules are there, and it has beta carotene molecules and some cytochrome B. Cytochrome B is also some 2 molecules are present inside. In addition to that, it has what we call quinones and phytophytin. So whenever you describe photosystem 2, it is composed of 6 important integral components. There some are polypeptides, some are reaction centers, and some are chlorophyll A and chlorophyll B molecules, and carotenes, and the other most important are plastoquinone and phytophytin. And of course, we have magnesium ions also. This magnesium, calcium, and chloride, these 3 are involved in photolysis of water. When water gets broken, you get protons and electrons and half molecule of oxygen. Now these protons and electrons, they move. So when 4 photons of light falls on photosystem 2, generally lower wavelength, 680 nanometers or so. So then this light, the photon moves very quickly. For its movement, it takes less than 10 minus 9 seconds. That is just billionth of a second. In such a span of lights, such a fraction of a second, the electrons from the photosystem 2 get excited, and this excited we denote by 6it asterisk. So now immediately this, so when it loses one electron, there is a deficiency of electron in photosystem 2. Now this deficiency is filled by with the photolysis of water. Now this photolysis of water is done by OEC complex, that is oxygen evolving complex, consists of calcium chlorides and manganese. These manganese calcium chlorides help in the photolysis of water. So 2H2O gets modified into or splits into 4 protons, half molecule of or 1 molecule of oxygen, and 4 electrons. So these electrons move from photosystem 2 and they are accepted by pheophytin. The initial acceptor of electrons is pheophytin. The pheophytin is nothing but a modified chlorophyll molecule wherein the magnesium atoms are replaced by hydrogens. Now this pheophytin transfers its electrons to quinones. Now from quinone, the quinone and absorbing electrons, it gets reduced. So acceptance of electrons is called reduction. Though the plastoquinone takes electron from quinone and protons from what we call stroma, and ultimately plastoquinone gets reduced to plastoquinol. Quinone becomes PQH2 with the absorption of electrons from quinone and taking protons from stroma. So protons from stroma, electrons from quinone or pheophytin, the quinone becomes quinol. So oxidized form is called quinone and reduced form is called quinol. So the PQ becomes PQH2 and PQH2 again becomes oxidized by losing its protons and electrons. Now the electrons are taken by iron sulfur protein. Iron sulfur protein that is present in cytochrome Bf complex. So the electrons are taken by iron sulfur protein and protons are released into the lumen of the thylakoid. Now these electrons from the quinone move to iron sulfur protein. From there they enter into cytochrome F. From the cytochrome F they move to plastocyanin, a mobile electron carrier. So here what is the function of cytochromes? Cytochromes accept electrons from plastoquinone and transfer electrons to plastocyanin, and photosystem 1 ultimately photosystem 1 extracts takes electrons from plastocyanin, and ultimately from photosystem 1 the electrons move to ferredoxin. Now coming to photosystem 1, this is operated by higher wavelengths. Its reaction center is denoted as P700. Photosystem 2 is denoted by P680. This is P700. Now, so from plastocyanin, electrons enter into photosystem 1. They ultimately move to ferredoxin. Now, there is an enzyme, ferredoxin reducing substance or ferredoxin reductase, ferredoxin NADP reductase, which taking electrons from photosystem 1 and protons from stroma, ferredoxin gets reduced or ferredoxin converts coenzyme NADP into NADPH2. Okay, so now if you observe very closely, I told you at 2 places. See, in the first oxygen evolving complex wherein 2H2O we have started, wherein 2H2O gives rise to 4 protons, 4 electrons and 1 molecule of oxygen. So these 4 protons are released into lumen of the thylakoid. Then with the oxidation of PQH2, that is quinol. Quinol gets oxidized to quinone, and here also 4 protons are released. So that way 4 plus 4, 8 protons are released, and with that, inside the lumen, the concentration of the protons is 1000 times more than that of stroma. But the thylakoid membrane is impervious. That is, it doesn't allow protons to escape across its membrane. So here, there is a small motor. Its details were given by Peter Mitchell, chemiosmotic theory. It is otherwise called ATP synthase or coupling factor. It has 2 parts. One is a stalk, the other is what we call head. The head is projected into the stroma and the stalk is within the membrane. Now, there is a channel. There is a channel through which the highly energized protons tend to move one by one. So, when 4 protons move across the membrane, through stalk, through the head, then 1 ADP becomes 1 ATP. That is, for synthesis of 1 ATP, we need 4 protons. So for the formation of 2 ATPs, we need 8 electrons. So that way, at the end of, so here what we have studied, we studied about photosystem 2, we studied about the electron carriers, that is quinone cycle we studied, and also cytochrome BF complex we studied. We studied about photosystem 1, then we also studied about the ATP synthase or coupling factor. So if you look at very closely, what is the ultimate end product? Ultimate end product is when electrons move from photosystem 1 to NADP oxidoreductase, we are getting reduced form of coenzyme, that is NADPH2. Then with the movement of electrons through what we call through ATP synthase, there is a production of ATP. This production of ATP from ADP is called photophosphorylation. So thus the end products of light reaction are ATP and NADPH2. These ATP and NADPH2s are called assimilatory power. These 2 energy-rich molecules are ultimately used in the reduction of carbon dioxide into carbohydrate. That what we call dark reaction. This dark reaction we'll study tomorrow. Now, any doubt? If you have any doubt, you can ask me. You can raise your doubts, or else we can call it a day. As far as the examination is concerned, these light reactions and also whatever we are going to study tomorrow, that is fixation or reduction of carbon dioxide through 3 important processes. One is C3 cycle, C4 cycle, and CAM pathway. That is reduction of carbon dioxide to carbohydrate. We'll study tomorrow. That is what we call dark reaction. So this dark reaction doesn't tell you that it occurs in dark. That is, its name dark reaction comes from, it is light independent. It doesn't need light, but the end products of light, that is ATP and NADPH2, are utilized in the reduction of carbohydrate to. So in photosynthesis, the photosynthesis, broadly speaking, oxidation of water and reduction of carbon dioxide. That is photosynthesis. So in light reaction, water is oxidized to protons and electrons, and as a result, you get ATP and NADPH2. And this assimilatory power is used in dark reaction to reduce carbon dioxide, to convert carbon dioxide into a carbohydrate, that is nothing but glucose C6H12O6. So this is that is also a complex phenomenon. Of course, we'll study about that dark reactions tomorrow. So if you don't have any doubt to raise, Now, I'll call it a day and hope to see you tomorrow, same time. That is from 3 to 4. Thank you.